In this video, I'd like to review how to conduct a dihybrid cross. I'm using in this example a parental generation of homozygous dominant RY and homozygous recessive RY. Remember that the first step in a cross is to find your gametes. And so for this individual, when meiosis proceeds, remember that meiosis reduces the amount of genetic information by half. So if I consider this first gene, the R gene, I'll only pass on one allele of that gene in meiosis. Because both of those alleles are the same, I know that it will give all offspring an R in each gamete. And the same for the second gene, it will give all offspring a dominant Y for that gene. So all of its gametes will have the same genotype for these two genes. The same pattern will hold for our second parent. It will give all of its offspring a recessive allele of R and a recessive allele of Y, because that is all that it has to give for these two genes. So when these gametes are combined, we will get the genotype heterozygous R and heterozygous Y. And this will be our F1 generation. So now that we know our F1 generation, what happens if we cross these two together? Again, we have to begin with the gametes that this genotype of organism can produce. So the pattern that I tell students to follow is to begin with the first allele. So I can give one allele of the R gene, let's say it's dominant R, and with that R, I can either give a dominant Y, or I can give a dominant R and a recessive Y. Alternatively, we could produce gametes with a recessive R and a dominant Y, or a recessive R and a recessive Y. So unlike the last example, when all the gametes had the same genotype, now we're producing 4 types of gametes. So that will allow us, because we figured out what we can use this to construct our Punnett square. Okay. Now, you don't have to do these in this order, but my recommendation is that you pick a pattern and stick with it, because then you're less likely to make mistakes every time you do one. You'll be following the same pattern. So just like we did with a monohybrid cross, we're just going to fill in the gametes. And by convention, I'm going to write the alleles for the same gene next to each other. So in this first box, I get 2 big R's and 2 big Y's. And I can proceed through all the boxes, just filling in the labels from each side. Now again, you'll see whenever I encounter a heterozygous genotype, I typically write the dominant allele first, regardless of whether or not it's on the top or the side. This again is just convention. If you wrote little r, big R, genetically that means the same thing. Again, some of it is just about the pattern, and if we always see it the same way, it makes it easier for us to understand. Almost done. So now I've completed my Punnett square for my dihybrid cross. And on this you can see that there are different combinations, and you can find the numbers that have the dominant trait for both, which would be 2, 3, 4, 5, 6, 7, 8, 9. Okay, and the way I'll often represent that is a big R blank, it's a big R blank, and a big Y blank. Meaning that this, it doesn't matter what the second allele is if I have a dominant allele. So I have 9 in that phenotype category. I'll find that there are 3 of the genotype category R blank homozygous recessive Y. So those are shown, switch colors, here, here, and here. So I had 3 of those. Then I'll see that there are also 3 that are homozygous recessive for R and have at least 1 dominant allele for Y. Those are here, here, and here. And then in my last category, I'll see that I have only 1 that is homozygous recessive for both genes. And so this 9:3:3:1 ratio is always observed from a dihybrid cross.